Welcome to all B.Sc. second year students in online lecture of Fishery Science, paper 6th, new paper from this year, Fish Disease Management, from which various fish diseases we are studying in last many lectures. In these various pathogens, the infectious pathogens, that is the crustacean pathogens or the crustacean parasites, in which first crustacean parasite which is to be we have studied in last period, which is called as fish argulus or the fish louse. Then next crustacean fish parasite, next crustacean parasite attack on fishes and cause the fish diseases. That crustacean parasites, that is the Lernaea, and the disease which is to be caused by the Lernaea crustacean parasite, that disease is called as the Lernaeasis disease. Lernaeasis disease, which we have studied today. Lernaeasis or the Lernaea disease. This Lernaea disease or this Lernaea crustacean, these are also called as anchor worm. Lernaea crustacean parasite, Lernaea crustacean pathogens, it is commonly or also called as, that is the anchor worm. Name itself indicate that, name itself it is to be suggested that the worm, the parasite, the crustacean animals which is to be anchorage, the animals which is to be anchor their body, anchor their head with the help of piercing organs on the body of the fish, that may be at the place of fins, on the skin, on the mouth or on the head. That's why this parasite, it is commonly called as the anchor worm. Commonly called as the anchor worm or the Lernaea parasite or the Lernaea disease. This is a crustacean parasite that cause the disease in fishes. This is a crustacean parasite that causes the disease in fishes, which is called as Lernaea disease. With the help or the disease which is to be occur, the fish which is to be infected due to the crustacean parasite, and disease is to be known as the Lernaea disease or the Lernaeasis. It is also called as anchor worm. It is also called as anchor worm. Is one of the most common ectoparasite copepods. It is one of the most common ectoparasitic copepods. It is the copepod crustacean. It is the copepod crustacean copepod parasite, and it is to be ectoparasite. Again we have studied ectoparasite and endoparasite. Ectoparasite, the parasite, the pathogens, the organisms which is to be grow externally on the fish body, external part of the fish body, and they can suck the blood, they can suck the tissues of the animal. These are called, they can feed the animals, they can feed on the bloods and muscles of the host animal. That parasite, it is to be called as ectoparasite. This Lernaea is the ectoparasitic copepod. Lernaea is the ectoparasitic copepod, and it is to be cause the fish diseases of freshwater fishes. Those fishes which is to be cultured in freshwater medium, those fishes, these are to be grow in freshwater aquatic medium such as pond, lakes, rivers, or in culture tanks, culture areas, in freshwater medium. The parasite, these are to be attacked, parasite, these are to be affect it is called as the Lernaeasis or the Lernaea disease. That is, this is the ectoparasite. Ectoparasite. This disease is caused by crustacean parasite Lernaea species. This disease is caused by crustacean parasite Lernaea species. Lernaea is commonly known as anchor worm. Lernaea is commonly known as anchor worm. This parasite is very destructive to the goldfish. It is most destructive, means abundant or maximum scale, on large scale. Those goldfish, these are to be cultured. Those goldfish, these are to be grown in either aquarium or those are to be grown in culture ponds, culture tank. This parasite, Lernaea parasite, is very destructive to the goldfish and the carps. Common species of Lernaea is as Lernaea elegans, Lernaea categories, Lernaea polymarpha. These three parasites, these three pathogens from the Lernaea crustacean. Which are these three pathogens? That is, these are most destructive in carps and goldfish. And common crustacean parasite, common crustacean pathogen, that is Lernaea. In this Lernaea, Lernaea elegans is a species, Lernaea categories is another species, and Lernaea polymarpha is the another species. These three species, these are to be attacked, these are to be caused the infectious condition, these are to be caused the infection of goldfish and carps in culture of fresh waters. These are to be more destructive, these are to be more dangerous. It is to be attack complete crop. See in the figure, the first figure, that is in this figure, see this is the fish, and this fish, these are to be affected on the body of this fish, on the skin. The parasite, these are to be attached. Parasite, these are to be affected. Which are these parasites? See, this is the parasite, this is the parasite, this is the parasite. These parasites, these are to be affected, these are to be attached on the skin of the body of the fish, and fish becomes diseased. In second figure, in this is the, that is Lernaea parasite, which is to be look like. This is the mouth, these are the host body, and with piercing organ, anchoring organ, organs, that is mouth is to be anchorage. They pierce their mouth, pierce their head inside the skin, inside the body of the fish, and they can then hang their body outside. These are the two egg capsules specially. These are the two egg capsules, these are to be seen, and when this animal, these are to be see outside the fish body, hanging tuft like structure, hanging the outer side of the skin, then we can identify, then we can recognize fish become infected due to the crustacean parasite. Fish become, these are to be affected due to the crustacean pathogens, and that crustacean pathogen is nothing but that is the Lernaeasis. That may be Lernaea elegans, that may be Lernaea polymarpha, or that may be another Lernaea species. With these three species, these are to be affected, these are to be caused the major carps as well as the goldfish, their crop, these are to be completely vanished by these Lernaea species. See, this is the figure again, another infected fish due to the anchor worm. Infected fish due to the anchor worm. See the fish, that is the goldfish, Cariaceus auratus, aquarium fish, as well as it is to be cultured in freshwater ponds and lakes. These fish, these are to be affected, badly affected or badly infected by the crustacean parasite. It is called as anchor worm, that is Lernaea. On whole body, number of Lernaea parasite, number of Lernaea crustaceans, these are to be affected, these are to be infected. Which is to be, we have seen here, first on the head region, this Lernaea parasite anchor, then in this is another Lernaea, this is another Lernaea, this is another Lernaea. These Lernaea, these are to be affected, these are to be seen. This is another Lernaea. These different, or specially these are the Lernaea parasite whole body these are to be covered, affected due to by the a green circled areas which is to be caused by the pathogens crustacean parasite infected fish due to the anchor worm, that is the Lernaeasis or the Lernaea elegans or the Lernaea polymorpha, which is to be caused, which is to be affected. See the fish, that is the goldfish. See another fish again, another fish again. Again this is a aquarium fish like that of the Rasborini family, and this aquarium fish again these are to be infected, these are to be affected. First fish arbulas these are to be attacked on the body, and then this fish arbulas these are to be fall down, areas where scale these are to be dropped down, and red patches these are to be appear on the body. These red patches these are to be seen, these red patches these are to be seen, and at the place of these red patches anchor worm or the Lernaea, this is the Lernaea or this is the anchor worm. These anchor worm these are to be infected, these are to be affected, and fish becomes at a time, it is to be multi-parasite, multi-pathogen, more than 1 or 2 or 3 pathogen these are to be affected, and fish becomes these are to be diseased. First parasite this is the, that is the crustacean fish arbulas or the fish louse, and this is the second, that is the Lernaeasis. These are the Lernaea. These two crustacean parasites these are to be affected, these are to be infect these fishes in freshwater areas, or these fishes which is to be cultured in ponds and lake. It is the infected fish. Again another fish of this Lernaea which is to be observed, which is to be seen. See the in figure again. When with the help of this piercing mouth these are to be pointed, and these are to be anchor their head inside the skin of the fish, and they can pierce, they can enter their head inside the skin of the fish, and whole body these are to be hanging outside the remain. This is the another parasite. See this parasite these are to be seen. This is to be look like sucker like structure, pointed sucker these are to be sucked. First they are enter inside the skin, and then they can suck the blood, then they can suck the tissues on which they can feed, on which they can be survive, and this is the animal there. This is the ovaries these are to be hanging outside to these such bifurcated regions, 2 lobes these are to be seen, and this is the body of this parasite which is to be observed, which is to be seen in this figure, in this insect. That is, it is to be infected by the Lernaea or the Lernaeasis. Again, this is the goldfish. Again, we have seen, we have observed how these goldfish these are to be infected, how these are to be infection these are to be seen. See on this area, these anchor worm these are to be affected, these anchor worm these are to be infect, and fish become diseased, fish become affected due to the, that is crustacean parasite. Hence, it is the more destructive and more dangerous for the culture of these fishes, these areas. It is red like external parasite which attaches itself to the host males and do not attack the fish and are unspecified or unspecialized for the parasitic life. It is red like external parasite, which is, it is red like external parasite which is attaches itself to the host males and do not attack the fish, that is females, and are unspecialized for a parasitic life. The female parasite itself attaches to the fish anywhere on the body. The female parasite itself attaches to the fish anywhere on the body of the means of anchor like appendages present on their head. The anterior portion of the parasite is buried deep into the skin of the fish or fin of the fish, and only the posterior region may be visible externally. Only the posterior, anterior part these are to be deep penetrated, anterior head region these are to be deep buried into the skin of the host parasite, that may be fish or that may be other animals, and remaining body these are to be externally hanging, visible to the naked eyes. This parasite affect the fishes particularly during the summer months. When that is temperature, these are to be more. When water temperature, these are to be increased, and water level, these are to be decreases. Generally, this condition, these are to be observed, these are to be seen in summer period, in summer months. This parasite affect the fishes, particularly during the summer months, when temperature, these are to be more, when temperature, these are to be maximum. At that time, at that period, this parasite, these are to be infect, this parasite, these are to be affected on the, that is, major carps. Then which are the symptoms of this Lernaeasis? Which are the symptoms of this disease? It is important, it is to be interesting to identify, to know the symptoms. Which symptom these are to be occur on the fish? The first symptom which is to be seen, infected fishes are by small holes and wounds on their body. Due to the Lernaea parasite, due to this Lernaeasis, those fishes, these are to be infected due to the Lernaea. Infected fishes are by small holes or wounds on their body, on the body of the host, on the body of the fish. Small wounds and holes are seen. When we have observed, when we have carefully seen, holes and wounds are seen on the body, it is the first symptom, that is crustacean parasite may be affected, crustacean parasite may be injured, crustacean parasite may be infected to the our culture fishes, our culture crop. Second symptom, the wounds also allow the fungus and bacteria and fungi to flourish. Those wounds, those holes, these are to be appear on whole body. In these wounds, in these holes, in these wounds, these holes, fungus and bacteria, these are grow ultimately. Just I have told to you. What is I have told to you? When holes or wounds, these are to be appear on body, multi-parasitic condition, these are to be observed, and it is to be more difficult to control, because the treatment, these are to be different treatment, these are to be separate for the crustacean, separate for the fungus, separate for the bacteria. When we have performed the fungal treatment, then crustacean and bacterial, these are to be increased. When we use the treatment of bacteria, fungal and crustacean parasite, these are to be well flourished. Or when we have used the crustacean treatment, then at that time fungal and bacterial pathogens, they can grow, flourish rapidly. Means it is to be more difficult and more dangerous to control such condition when fish, these are to be affected, when fish, these are to be affected or infected due to multi-parasitic condition. And in this condition, in this stage, the wounds also allow the fungus and bacteria, fungi and bacteria, these are to be flourish. It is to be more dangerous, it is to be more serious for the culture condition, for the culture ponds. Next symptom, the fish show slow growth and loss of balance. Due to this crustacean parasite, due to this infection of the crustacean, fish growth, these are to be slowed down. Development of fish, these are to be lost. Development of the fish, these are to be slowed down, minimized down, and loss of balance, these are to be occur. Due to this crustacean, due to this condition, fish balance, these are to be collapsed, and once fish balance, these are to be collapsed, these are to be lost, then this is the sign when fishes, these are to be not grow, and abnormally they can behave, abnormally they can lose their balance, their behavior, these are to be abnormal. It is the sign, it is the symptoms of Lernaea, or that is crustacean parasite Lernaeasis. Other symptoms, fish grows thinner, and their market value is greatly reduced. Suppose when fishes, these are to be caught, these are to be captured by fish culturists or the farmers, and fish is to be not grow very well. It is to be thin and thin, weak and weak. Ultimately market values, these are to be fall down, market values, these are to be reduced, because in market those fishes, these are to be large, well developed, their muscle bundle, these are to be fully developed, flesh, these are to be more maximum on the fish body. Such fishes, these are to be consumed by consumer, and they can purchase by the consumers. Suppose fish, these are to be not developed, muscle these are to be not developed, and fish these are to be thinner, look like a ugly condition. Such fishes these are to be not purchased by any consumer, and ultimately the market values these are to be reduced, and when market value these are to be reduced, it is to be harmful, it is to be dangerous for the fish farmers or the fish culturists. It is another a economical stress, economical symptom which is to be seen. Next symptom, the female carries 2 egg sacs. The female carries 2 egg sacs at hand end and can be easily seen by the naked eyes. When female parasite these are to be attack on the fish, especially the lobes of the ovary, lobes of the egg sacs. These lobes of the egg sac or ovary these are to be hanging outside the body of the host, and these are to be seen in 2 different lobes. And when female carries 2 egg sacs at hind end and can be easily seen when we have observed, when we have seen in our naked eyes, which is to be we have seen in first figure. See in this figure, these 2 lobes these are to be observed, these are to be seen. We have again in this fish or in this figure we have observed these are the 2 egg sac. This is one egg sac, this is another egg sac. These 2 egg sac these are to be carried by female, and when female these are to be infected, when female these are to be affect to the goldfish or the carps, this part these are to be hanging outside, and when we are observed to the fish these are to be seen to our naked eye. This is again another identifying symptoms for to identify the crustacean parasite or that is the Lernaea parasites. These are the few symptoms which is to be have seen up to. Which are these symptoms? Again I can summarize. Infected fishes are by small holes and wounds on their body. The wounds are also allow the fungus and bacteria. At time bacteria, fungi these are to be grow, flourish in these holes and wounds. Then fish show growthness is to be slow and loss of balance, abnormal behavior. Fish grows thinner and thinner. Flesh is to be not developed on the fish body. Ultimately market value these are to be reduced. Market these are to be lost. Again crop is to be lost, and when female these are to be affected on the host, this female these are to be carries 2 egg sac. Depending upon these 2 egg sac, when we observe the parasite, observe the infected fish by our naked eyes and seen the 2 lobes, 2 bubble like structure, egg sac like structure on externally, then we can confirm that fish is to be infected, fish is to be parasite affected due to the crustacean parasite, that is the Lernaeasis or the Lernaea elegans. These are the some symptoms which is to be have seen. Then next part, treatment of this Lernaeasis or this crustacean disease. Which are the treatment which is to be done? Our crop is to be infected, our area of culture fishes these are to be affected by Lernaea, by crustacean parasite. Then to minimize, to cure this disease, which treatment which is to be we have done, we have should perform. In this treatment, few treatment we have seen, few treatment we have studied here. The first treatment, dip the infected fishes in a weak solution of potassium permanganate for 2 to 3 minutes. It is the first treatment which is to be recommended, which is to be essential. Which is the first treatment? Those infected fishes that may be carps or goldfish, those are to be affected, those are to be infected by the crustacean parasite, that is Lernaeasis or the Lernaea elegans. Such infected fishes these are to be collected, these are to be selected, and these collected fishes these are to be deep into the weak solution of potassium permanganate for up to 2 to 3 minutes. When these infected fishes these are to be deep in such permanganate solution or the potassium permanganate solution for 2 to 3 minutes, these parasites these are to be paralyzed and they can fall down, or sometimes such fishes these are to be collect in hands. These parasites, these are to be paralyzed. Within short time, we can remove these paralyzed parasite. The parasite is first paralyzed by potassium permanganate or copper sulfate solution, or that is by a common salt solution, and then these paralyzed parasite, these crustacean parasite, then these are to be removed with the help of the forceps, by mechanically, by manually. But this is the more time consuming and more it is to be crucial, because every crustacean, every parasite, it is to be not properly removed and time being consuming, and fish is also in danger condition. For 5 minutes or 2 minutes it is to be outside the water, it is to be again danger condition. That's why it is to be not better, but it is another way, another treatment. The parasite is first paralyzed and then removed by the forceps. It is to be recommended, it is to be used. Or another treatment, these are to be done. The pond can be disinfected by KMNO4 solution. The ponds can be disinfected by KMNO4 solution. Again, those ponds which is to be infected, those water bodies, those are to be affected due to the crustacean parasite. Such ponds, these are to be disinfected by spraying a solution of potassium permanganate, that is KMNO4 solution. And when we can use KMNO4 solution, ultimately that water is infected and parasite pathogen can be die or they can be removed. Or another treatment, applying linden. It is a chemical, applying a linden at the rate of 8 ml per 1000 liter. When we can use a chemicals to apply to cure the Lernaeasis disease, that chemical is the linden chemical, and this linden at the rate of 8 ml per 1000 liter. 8 ml linden, these are to be dissolved, mixed into the 1000 liter water, and in such water these infected fishes, these are to be stored or the deep up to 2 to 3 minutes, and then these are to be removed. This parasite, ultimately it can be controlled, it can be fall down or removed. With these some treatment, these are to be seen. With this some treatment, these are to be seen here. First, potassium permanganate for 2 to 3 minutes. Then these are to be removed with the help of forceps, by manually, by physically, or the water bodies or the ponds or the lakes or the tanks, it is to be disinfected with copper or the potassium permanganate solution, or applying the linden at the rate of 8 ml per 1000 liter. These are to be applied, these are to be used. These few treatments, these are to be done. Then also these parasites, these are to be not controlled. Then infected fishes bath in 500 ppm of formalin solution. Linden is to be used, parasite is not controlled. Potassium permanganate is to be used, parasite is to be not controlled. Then water bodies, these are to be disinfected, again it is to be not controlled. Then next treatment, infected fishes bath in 500 ppm of formalin solution for half an hour. For half an hour, followed to this 5 minutes bath in 1:1000 glacial acetic acid. First, take these fishes bath in 500 ppm of formalin solution for half an hour. Half an hour, these infected fishes, these are to be deep into formalin solution of 500 ppm. And thereafter, after half an hour, these fishes, these are to be removed, and then these fishes, these are to be bathed into 1:1000 glacial acetic acid. When this combined dual treatment, these are to be done, performed by fish culturist, fish farmers, definitely this crustacean parasite, crustacean pathogen, these are to be removed, these are to be completely removed from the infected fish body, or these parasite can be killed out. Or another treatment, mechanical removal of the parasite, that is with the help of various chemicals. Mechanical removal of the parasite, that is with the help of various chemicals, applying on the various concentration, various chemicals such as linden, various chemicals such as potassium permanganate or other different antibiotics, other different chemical. These different chemical, they are different proportions, different concentration. They can apply, they can mix, and mechanically these crustacean parasite, these crustacean pathogen, they can be controlled, they can be treated. With this treatment, these are to be done, these are to be seen, and with applying all these above various treatment performed by fish farmers or the fish culturists, definitely crustacean parasite, crustacean disease, these are to be minimized. And when we can apply all these chemicals, that may be linden, that may be potassium permanganate, that may be formalin, or that may be other different chemical, these are to be applied, that may be crustacean, these are to be fall down, crustacean, these are to be killed down, as well as fungal, as well as bacterial, or any pathogen which is to be affect on the fish body, that pathogen can be paralyzed, that pathogen can be removed from the fish, and fish is to be free from disease condition, and we can save our culture crop, we can save our culture water bodies from these parasitic infections, these parasitic diseases.